Today. I actually started off as a public health veterinarian in the United States Public Health Service, working for the Food Safety Inspection Service. So I'm delighted to be here today addressing food safety, its intersection with biotechnology, and taking a look ahead at where this is going. To do that, I want to take a brief look at the history of food safety, how that led up to the introduction of biotechnology into production agriculture, and then looking to the future to see what's coming. Because as was just noted, we know that the global demand for food is only going to increase. You may think this is a graphic of global population increase. It's not. This is actually a graph of the global mean temperature and carbon dioxide. So there are competing challenges, conflating challenges that are coming forward, and we're all looking at new tools on how to address this. I do want to be very clear. Biotechnology is not in and of itself the answer. It is one tool, albeit a very important one, that we want to add to that toolbox so that farmers and people throughout the food production and distribution chain can utilize that tool if it fits in their circumstances and what they're trying to do. Now, I said I want to start with a brief history of food safety as well as genetic improvement of animals and crops. And I don't mean to insult your intelligence. I know that many of you in this room know far more about the history of food safety than I do. However, I do want to put it into the context of looking at the products of biotechnology and how those two things have come together to get us where we are today. As you know, prior to the 21st century, the history of food safety was really very local, very context specific. Generally, throughout human history, people consider food to be safe because it's what they eat. It's what their ancestors have eaten, their family ate, that they see other people eating. There really wasn't a standard definition of what a safe food was until very, very recently. Wholesomeness was the de facto and very localized food safety system. And that worked fairly well for a long time. You know, there are histories, for example, in Europe of laws requiring animals to be walked into a town or city before slaughter. That was a wholesomeness check to ensure that dead animals weren't being served to people. And again, this system worked fairly well. And an interesting thing to think about is that it also actually accounted for foods with known hazards. There are foods that people routinely eat that if they're not produced, prepared in exactly the right way, can be toxic. We've adapted to work with that. Concurrently with all this happening, there was a lot of movement in terms of the domestication of plants and animals to produce those foods that we eat. And we can see this, for example, looking at corn or maize from its wild ancestor to the corn that's grown today. Fundamental changes in that corn plant were achieved through the process of domestication, selection, and crossbreeding to get to where we are today. And that changed a lot of components of that plant. In terms of its structure, the composition of the product, the way it utilized nutrients, the places it could be successfully grown, tremendous progress there. We see this in animals too, for example. I chose Scotland. I could have chosen anywhere on Earth, really, to show the heritage and history of animal breeding and how it led to incredible variation within a species. So Scotland itself has almost 10 different recognized breeds of cattle that all have very different characteristics and were intended for very different purposes in very different regions. And that was all done through natural selection, if you will, through controlled breeding, crossbreeding, protection, development of specific strains. So a lot of heritage in that as well. We get to the 20th century, though, food safety starts to become standardized. The jungle obviously spurred a lot of that in terms of the meat industry, but really this shift to preventing foodborne hazards, right? Whether they be pathogens or other harmful microbial or physical contamination. And as was indicated earlier, that led to the development of the HACCP system and a lot of innovation in terms of making sure that food is safe, that it's brought to the consumer in safe condition, that it's free from adulteration and contamination. Also in the 20th century, there was a lot of new technology that was brought to bear in terms of particularly plant breeding. To date, animal breeding still pretty much remains one animal crossed with another. But on the plant side, we saw a lot of innovation. And I know it's a big audience, but just a poll, how many people know what this is a picture of? Anybody? This is a picture of an old radiation garden from the 1960s. Okay? This was, for a while, very popular, presented to the public as an exciting new benefit of the nuclear age. And essentially what you're looking at, this particular one is no longer used, but that's a small nuclear reactor in the middle of that picture. And you would plant different plants or crops at various degrees of separation from that reactor, expose them to radiation, and see what sort of mutation you could induce in the plant. And from that, you could select new traits, new characteristics that you would propagate. And believe it or not, this remains a way that plants are bred to this day. Induced mutation, kind of taking that natural mutation that occurs in any mating event and really accelerating it through the use of radiation to produce a lot more variation. And this really has been used in most of the foods that we eat today. One of the most famous examples is the pink grapefruit, or the ruby red grapefruit. This is a result of radiation breeding to get a grapefruit that will maintain that bright red color after it's picked, harvested, and brought to the consumer. Now, of course, the plants themselves are not radiated constantly, but back in the ancestry of that product, it was a result of this mutation breeding. I won't insult your intelligence by telling you why radiating animals has not been as successful. But, again, a lot of science and technology has gone into this, and that really led to the development of a lot of our hybrid seeds that have really revolutionized agriculture to date. And that brings us to now. When we talk about biotechnology. Now, I'm using the word biotechnology, but really the rest of the world uses the term GMO, or genetically modified organism. How does that fit in to agriculture today? What has happened with that? There's a lot of different competing definitions out there, but I'd say most of the world, from a regulatory perspective, uses the definition of actually living modified organism that is found in the Cartagena Protocol. And what that basically says is that a living modified organism means any living organism that possesses a novel combination of genetic material obtained through the use of modern biotechnology. Modern biotechnology is basically defined as including recombinant deoxyribonucleic acid, or DNA, rDNA, into a cell. This is all a very fancy way of talking about transgenic applications, where you're moving a gene from one species, organism, to another in a way that could not occur in nature. So what does this really mean in practice? To produce a farmer-ready product through biotechnology, you have to be able to introduce that new gene, that new trait. And this is actually fairly difficult to do prior to the advent of genome editing. Don't have time to go into all the science behind it, but basically there's two primary techniques that have been used, either utilizing a bacterial vector or actually gunpowder to drive DNA into plant tissue. It's a random insertion. It's difficult to actually make that happen. You've got to get that in. The next thing that you have to make sure is that once you get that gene from another species into the plant or animal, it has to be expressed, right? Putting the gene in there, in and of itself, is not going to have any benefit unless the plant or animal is able to read that gene and use it to produce the protein that expresses the result that you want. So that's another critical hurdle. And finally, and I'm sorry, all one, not 1, 2, 3, but the third important hurdle that you must overcome is that that insertion must not have any negative effect on the plant or animal, or prevent it from doing anything that it needs to do to grow or thrive, or present another hazard. So, 3 critical bars that you have to reach using this older technology, again, prior to the advent of genome editing, to make something that works. And that takes a lot of time and investment. We'll get back to that in a moment. But here's an example. This is one that's very, very commonly grown in the Western Hemisphere, and that's Bt corn or maize. And what this is basically, it's called Bt corn because they took a gene from Bacillus thuringiensis, which is a ubiquitous bacteria found in the soil, farms globally, worldwide, not just farms, soil everywhere. And they noticed that there was a couple genes that this bacteria produced that had very promising, very strong effects against insects that were key pests of corn. These are all variations of a few genes that are specific to certain insects that have been identified, and through trial and trial and trial, added to the corn plant. And what it does, it expresses something that's called a Bt delta endotoxin that again is specific to very certain insects, with the result that the plant has enhanced resistance to, again, these insects, which tend to be caterpillars, colloquial, but larvae of Lepidoptera. And the 2 big key ones here were European corn borer and corn rootworm. There's a lot of words on there, and it probably sounds scary even of itself, but I want to show you this because, you know, a lot of times the public hears, as was mentioned earlier, frankenfood, something scary, like what's going on here? What's being put into these plants? So in this case, perhaps the most commercially successful application of biotechnology to date, we're talking about a few specific related genes from the bacteria commonly found in nature that when put into the corn plant allows it to be resistant to insects that are key pests for corn production. It was mentioned earlier this morning by Dr. Parberg, the dramatic reduction in insecticide use in US corn production. This is a primary driver of that. Here are a few more key examples of some of these transgenic applications of biotechnology. Another one, of course, is Roundup Ready soybeans. Very, very, very highly cultivated in the Western Hemisphere. And this, again, was a specific gene from Agrobacterium, another ubiquitous bacteria in the environment. This particular gene, when added to a soybean or corn or cotton or other plant, confers resistance to the effects of the herbicide Roundup. I realize that glyphosate use, Roundup, is a controversial topic, but I want to be clear that that not necessarily means that food safety needs to be a proxy for that war, okay? The insertion of this gene itself does not cause the plant to produce produce a herbicide. It merely makes it resistant to its effect. Another star, if you will, of biotechnology is the rainbow papaya. A couple decades ago at this point, in the United States, in Hawaii, our major center for production of papaya, that industry was decimated by the papaya ring spot virus. We almost lost that industry. However, they were able to save it by inserting some genetic material from the virus itself into the papaya, which conferred resistance to that same virus. So again, this small insertion was able to save that industry. Here's a few more that are not yet commercialized or hopefully will be soon, just to give you an idea of the fact that this technology is still being used and there are exciting things that are coming down the pipeline. First one I want to highlight is low methane rice. Obviously, when we're looking at climate change, looking at human drivers of climate change, how can we reduce that impact? Rice production produces a lot of methane. There has now been, through biotechnology, rice produced, still in the experimental stage, however, with the insertion of one gene from the barley plant that changes the way that the plant interfaces with the soil environment, the biome, the microbes, etc. in the soil, to reduce methane production. Another similar innovation is in wheat. HB4 wheat has now been developed and has been approved in several places, including it's under evaluation in the United States. This is a biotech trait that confers drought resistance. Okay? It changes the metabolic pathways by which the plant either decides to go into senescence or not when exposed to a deficit in water. In other words, simple terms, can make wheat more resistant to drought. And this